This is topic for exam 4, topic number 1, part 3. So the structure for the enzyme where the active sites are located consists of 3 subunits. And this is like the top-down view. Each one of these little circles is a C subunit. And the 3 of them together make a unit that you can purify, the catalytic trimer. These assemble as 2 units. So there's 2 of these, and we'll draw a sketch of what that looks like from the side. So there's a 3rd subunit behind here. Each one of these is the catalytic subunit, and there's 2 layers to this whole structure. So here's a catalytic trimer and another catalytic trimer. And the 2 of them, these 2 catalytic trimers, are held together by regulatory subunits, which are organized as dimers. And there's 3 of these, and we'll draw those in here. So here's, and then the ones that are behind are located there, and then here we have our, each one of these circles is an R subunit. What makes ATCase so special are these regulatory subunits, because they can change their shape and therefore influence the structure, the structural equilibrium between the T and the R forms of ATCase. The R form has a more open structure, and it is also the more active. It binds and interacts productively with aspartate, the substrate, easily. And one way to describe that is that the Km is relatively low. For the T form, it has a much more closed structure. It's less active, and it interacts with difficulty. Aspartate can bind only weakly, and we can describe that situation that the Michaelis-Menten constant for this form, if we could isolate it, would be relatively high. And the net effect of this structural equilibrium is that we observe cooperativity. If we plot the rate as a function of the concentration of aspartate, we get an S-shaped curve. It's very analogous to the situation that we saw for hemoglobin. And this curve can be further modulated by shifting the balance point between T and R forms. We can favor the R form by adding ATP. And in that case, we get a curve that's not quite as cooperative, but is more active. So this is with ATP. And we can also make it more difficult to achieve the R form by adding CTP, the other nucleotide. That's the basis of negative feedback inhibition. And so this is the curve that you would get with CTP. More cooperative and less activity for any given amount of aspartate.